Next, we have Tom Hanke, who's a professor of vaccine immunology at the University of Oxford and distinguished professor at Kumamoto University. He does work that spans basic translational clinical research into the development and testing of HIV vaccines, and he's going to talk to us today about targeting HIV where it hurts.
Hello, good afternoon. I would like to start by thanking for the invitation. It's nice to be here in this very nice place and an interesting conference. So, even in the broader context of increasing the antiretroviral treatment and prevention, HIV vaccine is the best solution and likely key to ending the epidemic. So our aim is to induce effective cytotoxic T cells. And the key word really is effective. These can complement broadly neutralizing antibodies for prevention, and they might be key for cure. What is clear, that more of the same CTL is not going to work. Natural HIV infection induces vigorous T-cell responses which don't protect. Past T-cell vaccines using full-length proteins, including the most conserved and promising GAG, didn't work. Just reactivating T-cells that failed to control HIV prior to antiretroviral treatment is not going to work because HIV has escaped them already. We just have to do something different. So the biggest challenge, both for T cells and for antibodies, is are the HIV diversity and escape. So there are various ways how to tackle diversity. One can use heterologous mixed immunogens, but can use natural sequences with the shorter distances to the group that you want to protect against. One can use artificial center sequences or maybe focus on transmitted from the viruses. There is a bottleneck for transmission, and maybe your life is going to be much easier if you can find out what's common to these viruses. Or one can compute these immunogens that I'm going to tell you more about. So T-cell responses focus on immunodominant epitopes, which happen to be most in the most highly variable regions of the HIV proteins, and are therefore easy to escape, and therefore are less protective. Approaches using whole proteins address the diversity problem only partially, because they do not redirect T-cell responses away from these decoy regions. So our strategy is to focus the T-cell response on the functionally conserved proteins, regions of proteins, sorry. These are common to most variants, and mutations in these regions are typically associated with replicative fitness loss. And back to the previous questions, epitopes are evenly, we think, distributed across the HIV proteome. So the conservation comes from function and structure. It doesn't come from the fact that these regions wouldn't be efficiently targeted by the immune responses. So our first generation conserved region immunogen is designed as alternating conserved clade consensus. These are the regions that we include, 3 regions in GAG. Most of the regions come from whole, with and 2 envelopes. For each region, we have used clade consensus, and we have alternated the clades so that we have equal we have alternated the 4 major clades so that we have equal clade representation. And of course we do preclinical studies, but I'm going to focus on the clinical development. We insert this immunogen into lots of different vectors, but the 3 that we tested in clinical trials is DNA, MVA, and chimp adenovirus. And these vaccines have been tested all together in 8 different clinical trials. The top 4 show the trials in HIV negative individuals for prevention, and the bottom 4 show trials in HIV positive individuals as a step towards cure. And I'm going to show you some data from HIVCO2, which is for conserved regions, and HIVCO4, and then I'm going to show you this Spanish study that I was asked to talk about. So HIVCO2 took part between March 2010 and August 2012, and there was an extension, and it tested 3 different viruses, DNA, MVA, chimp adeno, delivering these conserved regions. We had 3 arms. In each arm we had 8 vaccine recipients and 2 placebo, and we have tested chimp adeno prime and MVA boost, or 3 DNAs followed by chimp adeno prime and MVA boost, or 3 DNAs followed by MVA and chimp adenovirus. So first of all, 100% of vaccine recipients responded to the vaccination, and no response was detected in placebo. We have induced high magnitudes of spots, or of the HIVCO-specific interferon gamma producing T cells in the blood, and the 2 highest regimens were chimp adeno prime MVA boost and DNA prime chimp adeno prime MVA boost, and we have reached over 5,000 spots median peak responses. These immune responses last for at least 2 years, and in those volunteers who came back 2 years after the last MVA administration, the median ex vivo responses in the blood still circulating were 600 spots. So these frequencies compare favorably, and the frequencies of responders to the STEP study. Because we are using in natural infection subdominant immune responses, we take them out of the context of the whole proteins and of HIV, and we deliver them by potent vector regimen. We are actually inducing lots of new responses to lots of subdominant epitopes which weren't detected before. So just from this trial, we looked at about 10 volunteers, and we have identified 28 epitopes which were identified before, of which 14 we have identified in great detail, and for the other 14 they are in the so-called B list, where there is some missing information. So it's a very good opportunity for mining new HIV epitopes. So HIVCO4 was in negative adults in Nairobi, took place in 2014 to 2015, and the aim was to repeat the most immunogenic regimens in African population. We couldn't use the chimp adeno for this study because GSK took over Okairos, and they didn't allow us to use this now co-owned vaccine in Africa. Fortunately, I only had on the shelf at 35 with green, which is for GAG, reverse transcriptase integrase, and there was a very good match with our conserved regions, so we could have substituted our chimp adenovirus. And again, we had 3 arms, 20 vaccine recipients for placebos, adeno prime MVA boost, or 3 DNA prime adeno electroporated the DNA, followed by, I don't know, an MBA. All these studies are published. So, once again, 100% responders, all vaccine recipients respond to the immune responses. These are the 3 regimens. Here's the DNA. Normally, when we do intramuscular DNA, we have hard time to detect any immune responses. The electroporation improves this, but overall, there isn't much difference. And 24 weeks later, the frequencies of interferon gamma producing cells in the blood are very similar. Of course, that doesn't mean that maybe the DNA prime is low until produce higher affinity T cells, and these investigations are still.
Just keep buying it.
These are the frequencies, the median frequencies, peak frequencies again 2,000 to 3,000 telespots in each arm. What is the breadth of the responses? So we have 6 peptide pools across the entire immunogen. So this allows us to have the first glance at the breadth of the immune responses. These are the 3 regimens and responses to pool 1 to 6. The important part is here. The median breadth response to the 6th pool is 6. So broad immune responses induced by this vaccination. So this is interferon gamma ELISpots, which of course don't mean much. The most relevant assay that we can do in vitro before we do efficacy in vivo is virus inhibition assay. Virus inhibition assay comes in different colors and flavors. The way IAVI does this assay is that we expand CD8 T cells by bispecific antibodies. That is in antigen non-specific way. We expand target cells by bispecific antibodies, infect the target cells with the viruses. They put these 2 together and look at the virus inhibition in vitro, and we compare the virus without effectors growing in all of the CD4 T cells compared to when we add the effectors which can or cannot inhibit the virus growth. So as I said, this is the best we can do prior to efficacy in vivo. So IAVI has a panel of 8 viruses from 4 different clades. Some of them are lab adapted, some of them are transmitted under infectious molecular clones, some primary. They are either X4 or R5 from different places around the world. And here we're looking in green at the adenovirus prime MVA boost or DNA adenovirus MVA, or in blue is the electroporated DNA compared to placebo in white. And out of these 8 viruses, we have inhibited, or the T cells induced by our vaccination inhibited significantly more 6 out of these 8 viruses, and 2 viruses that didn't achieve significant inhibition. There is a still clear trend that there was an inhibition. So conserved regions delivered by our vaccine can induce T cell specificities that can inhibit 4 major clades of HIV. So this was the prophylactic setting. Now I'm going to show you the therapeutic vaccination. So the first trial towards cure is this Barcelona 1 and Barcelona 2 trials, where 24 patients were put early on R, including raltegravir, and they were stable for 6 months, after which they received chimpanzee and then being boosted either 8 or 24 weeks apart. Again, this is BCN1. We had 100% responders. Every vaccine recipient responded by HIV-specific, HIV-specific T-cell responses. These gives you the left graph gives you the frequency of responders to these individual peptide pools before in blue and after vaccination. So you can see that the vaccine induced really good immune responses, again broad immune responses, and this is the absolute magnitude pre and post vaccination. The median was 900 or over 900. So then this is BCN1 here. These volunteers, or 15 of them, were invited back to BCN2 where they received MVA, 3 doses of romidopsin and second MVA. The reason for having 2 MVAs is because there were some suggestions that romidopsin may inhibit CTL induction or activity. And then 8 weeks later, the antiretroviral treatment was stopped and the volunteers were put back on antiretroviral treatment if they have 2 consecutive viral load measurements above 2,000 copies per ml. So this is the immunogenicity again of BCN1. You can see we have induced immune responses, and the first 2-3 years later, the immune responses were down, but were quite nicely boosted with the first MVA, and then the second MVA boosted this even further. How about the refocusing of the HIV-specific T cells? So, remember, it's HIV-infected people, they got HIV-specific responses prior vaccination, and this would be 6% of their total immune responses are focused on the conserved regions present in the vaccine. Then when we put them on R, the immune responses go down, and then during BCN1, chimpanzee-derived MVA boost, refocus these immune responses so that half of the HIV-specific responses now can recognize these conserved regions. During BCN02, 2/3 of the HIV-specific responses in these volunteers are refocused to the conserved regions. We have observed that after the first MVA vaccination, 60% of patients actually had a detectable viral load in blood, and after romidopsin it was 100% reactivation. And overall, we looked at the decrease of the latent pool, and there was a decrease in the latent pool, but it was consistent with the time of antiretroviral treatment, and there was no extra decrease due to the treatment. So what happened when we stopped the antiretroviral treatment? So typically patients rebound within 4 weeks, and that happened in 8 out of 13 volunteers. While 5 out of the 13 volunteers controlled the virus without the treatment. 1 of them had to be put back on R at 8 weeks post stopping, but 4 of these 5 still controlled the virus 6 months later. So this is about 38 or 31% control, which would compare to the 10 to 15% of spontaneous control observed in the Visconti trial. Are there any correlates of protection? Of course, we got small numbers, and we cannot really see anything. Here we're looking at the total immune responses on the y-axis, and x-axis is the latent size of the latent pool. And the pink dots are the 5 controllers, post-treatment controllers, and there is no obvious association. There is a bit more clustering here when we look at the refocusing, what percentage of the volunteers' HIV-specific responses were refocused on the conserved regions, again against the bars latent virus pool. And of course, on the top of the fact that these were all patients early on ART and that they received Romidepsin, there seemed to be some clustering to those people who had good refocusing of the immune responses and low virus load, low latent virus load. So the conclusion, we didn't see any reduction in the latent reservoir beyond what it is expected from being ART. We observed some signal for the ART-free control, but these are very preliminary data. Of course, we got no controls for mature control rates. It was an open-label study, non-randomized, and limited sample size. This is basically a hypothesis-generating pilot study. We have also ongoing river study where 60 patients at 6 UK centers were early on ART, and 30 of them, half of them will receive or have received chimpanzee and MBA boost 8 weeks apart, and then treatment of morinostat. Here we not, in this particular study, we're not going to stop antiretroviral treatment. We are just going to look at the size of the latent pool. But we are, of course, now after the Barcelona study, want to go back to the partner and see if we can boost these immune responses later and do this monitor antiretroviral pulse. And I should acknowledge Steve for this lovely picture of kick and kill. So, first generation vaccines then demonstrated that these vaccines are safe and immunogenic, and they are safe and immunogenic in HIV negative and positive individuals in Europe and in Africa. And they have proved the concept and therefore support timely clinical development of the second improved generation T-Cons vaccine that I'm going to describe to you in the next part of the talk. Oh, lost one slide, right. So what is the concept that we have proven? The concept is that if we take subdominant regions or epitopes out of the context of the full proteins or the whole virus, we present them by a potent regimen, such as chimpanzee and MBA boost, even to humans. We induce strong immune responses that can control HIV replication in vitro in autologous CD4 cells of 4 major clades. So second generation, because of the GSK story, of course, we needed to have our vector, chimp adenovirus, and it was also an opportunity to rethink the insert. And so the concept hasn't changed. The central concept is to focus the immune responses on the conserved regions of HIV protein, common to variants, important for replicative fitness. We're not talking epitopes as bits on the string, as conserved epitopes. We're not talking full-size conserved proteins like egg. We're talking regions. This is further enhanced by using bivalent mosaic designed by Patrick Korber, which increases the perfect epitope match to global variants and also increases the breadth and magnitude, as we saw in the previous talk. On the top of that, we also included Christian Brenner's protective epitopes and also Japanese epitopes. So we have included as much as we could conserved and protective epitopes on 4 different continents. We have decreased the number of regions. One slightly annoying observation in the first in all 2 study was that the median breadth of responses was 10 epitopes and 20% of them were junctional. Remember, we had 14 regions, therefore 13 junctions. I think we were unlucky at that point because in Barcelona they specifically looked at junctional epitopes and they didn't see any responses. But based on this experience, we have reduced the number of the conserved regions. We have 2 regions in GEC, the whole P24, and 4 regions in POL. Betty has computed bivalent mosaic. These 2 mosaics differ in about 10% of amino acid. And if you look here at the conservation, so this is the PTE perfect match to global variants from 0 to 100%. We have achieved about 80% perfect match to all global variants. And so the more in red it's perfect match, orange is 8 out of 9 amino acids. And this is what is in the vaccine, and this is what's left out, and you can see it's much less red. And then the last thing is that, again, to decrease the junctional responses, we have reshuffled these regions into into 6 different orders, so that we actually boost the prime with each region only. We have decreased the dose of each junction for each immunization. Remember that mosaic 1 and mosaic 2 are always used together. And then we never boost with the same junction again. And it happened that the mosaic 5 and 6 is in chimp adeno, 3 and 4 is in MVA, and 1 and 2 is left for other vectors like DNA, RNA, and other vectors that we use, lentivectors and BCG and whatever we think about. So, in terms of delivery, we have chimp adenovirus prime and MVA boost, which was shown by Adrian Hill and Eleanor Barnes, and also by us, being highly immunogenic in humans, except that we now use ChAdOx1, which is fully owned Oxford vector. These are the vaccines just to visualize. For prime, we need to make 2 chimp adenoviruses. For boost, we need to make 2 MVAs with 1 mosaic and the second mosaic in different orders. We have 6 different proteins, so we have to show that all of them are expressed from the DNA, MVA, chimp adenovirus. And we have shown that the 2nd generation vaccine is equally immunogenic in mice to the 1st generation vaccine. So I'm showing you the 1st generation and then mosaic alone, mosaic 1 alone, mosaic 2 alone, and the 2 mosaics as we want to use it in the vaccination. Basically DNA alone or chimp adeno alone and MVA induces equivalent immune responses. Of course, the viewers may say equivalent is no good. You have to be better than your first generation. Well, the 1st generation induced the highest frequencies of HIV-specific T cells in the field. So I think that's a good start. And on the top of that, it's going to have all the advantages of the 2nd generation concept mosaic. So of course, when we mix the vaccines, we induce even broader immune responses. Here you can see we've done lots of these mixing and separate arms and things, and it's all published. And in our hands, it doesn't make much difference. In this case, for mosaic, it did make difference in monkeys for peptide boost. Importantly, we didn't induce any junctional responses in 2 strains of mice. Now, this is a very important experiment. So, we are claiming that we are refocusing immune responses to the protective regions of the HIV proteome. And therefore, if we go to treatment-naive patients, those patients who have responses to these regions should be doing better than responses that don't, than patients that don't have responses to these regions. And this is exactly what we have observed in Japan in treatment-naive HIV-positive individuals. I'm showing you data for 120, and here you can see when we look at the total magnitude against now 10 different pools, because we've got 400 peptides of 2 mosaics. So the stronger the total magnitude, the stronger, the more L spots, the more peptides these the stronger total magnitude against the second generation vaccine is, the lower the virus load, the stronger the magnitude, the higher the CD4 counts are. If you look at the breadth, we have 10 peptides, the 10 peptide pool, so it's either 0 or increasing breadth. The broader the immune responses to these peptides, the lower the virus load, and the broader the immune responses, the higher the CD4 count. If we take out patients with the protective Japanese allele, we are left with 77 patients, and while we lose the significance for the virus load, the more clinically important CD4 count still stays. And I can tell you that now we have data for 200 patients altogether and 147 without the protective alleles, and we have now reached the significance also for the virus load. And therefore, our vaccine has the potential to reach to the general population, not only to the people who are lucky to have the protective HLA alleles. And these are the trials that are already funded for the second generation. So again, we have trials in HIV negative individuals. We're going to do trial in Oxford, just to show safety and immunogenicity, and perhaps compare it to the Christian Brenner's Immunogen. And we have already funds from the European and developing countries clinical trial partnership to do a multi-site phase 2A study in Africa together with IAVI. We have also funds through the European Union to do small trials in Barcelona, approx Milano, in HIV positive patients, and there's money for 6 patients in David Margolis and Bar Haynes cohorts, and Nil Gunateleke is waiting to hear whether she will get money for this about 30 patient trial, where we're not going to stop antiretroviral treatment. This is all about characterizing the immune responses induced under antiretroviral treatment in these people. And I had other expressions of interest for HIV cure from these and other groups of investigators. And I leave you with few pictures from the fisher folk communities on Lake Victoria who have high incidence, and where we, through the EDCPP and with IAVI's help and with all the local teams, we are planning to carry the phase 2A study in high-risk populations and build capacity for phase 2B efficacy study in the future. And so, acknowledgement, this is the Oxford team and all other teams, lots of people involved in these clinical trial teams. Special thanks to volunteers without whom we couldn't progress our HIV vaccines, and special thanks to the funders. And I'm happy to answer questions. Steve is approaching.
So the Barcelona study that was presented in CROI, you spent a lot of time with this therapeutic vaccine to generate responses to these conserved regions that are difficult to generate naturally. So you do that. You show that nicely. So what I understand is, so you spend all this time and you generate these responses. Why then come back with romidepsin, which brings out the natural infection again, and presumably would just increase the dominant responses prior to interruption? Does that question, do you know what I'm asking?
Well, I know what you're asking, but I have to remind you about our concept. Our concept is that we focus the immune responses to conserved regions which are common to all viruses. So if we reactivate no matter which virus, including escape viruses. So our hypothesis is that if we reactivate these viruses, we can hit them precisely and efficiently. So it doesn't matter that and the reactivation, of course, is good if we want to achieve eradication. And of course, future study will separate and tease out all these treatments separately. And this was just a pilot study where we were trying to do as much as we can.
Yeah, no, I understand the pilot study, just to do everything that was possible, kitchen sink approach. Now we got to figure out what happened. So did the romidepsin, when you had the original virus population, did that shift the immunogenicity back to the immunodominant responses? Sorry for the interruption. Did you guys look at that?
So we are analyzing the viruses. We haven't done, this is all ongoing. We are sequencing the viruses and we are looking which feedback these people were at, and all these questions that people have are ongoing. But the main thing is, remember, conserved regions. We're focusing on conserved regions that are common to all viruses. So we'd love to reactivate all the escape mutants and everything because we know we can hit them precisely.
The reason why you want, the only way to get those conserved regions is to avoid the rest of the virus getting in the way. And that's,
Say that doesn't make sense.
in the natural infection, you generate responses to the you generate those immunodominant responses for which you get escape.
Yeah.
And those CD8 responses, you don't want them to be there because they're not useful. And I'm just concerned whether or not latency reversing agents in these, because now all these cure studies are going to be these vaccines plus latency reversing agents, and they're the natural infection again. So I'm just asking questions, but
I've explained already. Conserved regions are common to all viruses, including escape mutants and whatever we reactivate. So our hypothesis is that by targeting conserved regions, we can hit these viruses precisely and efficiently. And we want to flush them out if we want to deal with latent pool.